welcome. I'm Kelly Gannon, the New England Academic Account Manager at Cell Signaling Technology, or CST, and your moderator for today's webinar, No More Data Secrets, Unlock the Molecular Mechanisms of Pathogenic and Non-Pathogenic Th17 Cells. Today's speaker is Dr. Maj Arist, a senior scientist with the Single Cell Technologies Group at CST. Maj is a leading expert in single cell research and has led the development effort of the InTraSeq technology, a novel method for simultaneously measuring RNA and proteins in individual cells, including post-translational modifications. Without further ado, please welcome Maj.
Thank you, Kelly, for the introduction, and thank you everyone for tuning in today for the webinar. Like Kelly mentioned, my name is Maj. I'm a scientist at CST, and I'm going to tell you about our latest product line that we released, the InTraSeq. We released it a couple of months ago, and I'm going to show you in this webinar today how you can use this technology to measure proteins in your single cell assays and why this is important and how you can use it in your biology research. So today the talk is about a collaboration. I mean, some of it is a part of a collaboration with Dr. Vijay Kushru at Harvard Medical School, and InTraSeq is short for intracellular protein and transcriptomic sequencing. So for today's agenda, there's going to be multiple sections. The first one is quickly going over what single cell analysis is, and then talk about the InTraSeq technology, what exactly is the technology, and then go over the protocol, the benefit and proofs, and some of the data that I will show in this webinar, including the collaboration that we have, as well as the product offerings. And please stay tuned until the end, because we have some new products that just released today that I will talk more about in the last couple of slides, and some additional information that can help you get the most out of the single cell experiments. So what is single cell analysis? It's a technology that was developed around 2010, and for over a decade, it's been used in so many different research in biology, and it's used predominantly to identify hard to detect cells, especially if your cell of interest represent only a certain small population or subset in a very heterogeneous and complex tissue, which also has been used as a technology that was used to look at cellular heterogeneity, to understand the transcriptome and finding novel biomarkers in individual cells in a complex tissue, as well as understanding how different cells interact with their environment. It's been used to, like I said, study gene expression in different cell types, understands how disease develop and how certain drugs can impact certain cell populations and not others, and hopefully, in the future, help you in helping with the personalized medicine fields. Traditionally, single-cell RNA sequencing data sets is visualized in a UMAP, which is a two-dimensional plot that reduces multiple dimensionalities made from principal components. And each dot in the UMAP represents one cell, and each color here represents one cell type. And in this case, for instance, the cluster of blue cells represent the T cell, and the cluster of orange cells are monocytes. And you can see that these are peripheral blood mononuclear cells. And I want to mention something which will be relevant later in the talk today, that the closer the cells are to each other, the more transcriptionally similar they are to each other. And the further away they are from the UMAP, the more they have distinct RNA or transcriptomic markers specific to each cell type. Now, this is a schematic here looking at the cell, a single cell, and these outline the RNA molecules in your cells, and the circles outline certain proteins. And the reason why I mentioned that, because around late 2017 and early 2018, CITE-seq was developed, and it's a technology that's short for cellular indexing of transcriptome and epitopes by sequencing. And you can imagine why CST is interested in this technology, because now we're introducing antibodies. And we at CST make a lot of different antibodies for so many different proteins, and specifically also for signaling pathways, hence in our company name. So when CITE-seq was developed, the researchers conjugated these antibodies with the DNA barcode, and they stained these single cells with these DNA conjugated antibodies. And then after the cells were washed, so you can get rid of the unbound antibodies, this complex and many other cells were loaded in a single cell platform where the single cell technology will capture the RNA from individual cells as well as capture the DNA from each antibody. And because through this technology you can quantify how many transcripts there are, you could also quantify how many antibodies are bound to your cells through the DNA barcode. And you can imagine that we at CST, we want to leverage that technology, the DNA barcode and antibodies, and allow you to measure all of these intracellular proteins, including cytoplasmic proteins, nuclear proteins, and all of these signaling pathways that can help you understand why certain proteins are active and inactive in a single cell RNA-seq format. Now, when I joined CST almost 4 years ago, we realized that in order to achieve that, you have to fix and permeabilize the cell. And this is outlined here when you have these holes in your cellular membrane. And we found certain challenges that we did not expect. And one of them was that the transcript is leaking out of the cell, which results in the loss of RNA. And you can see that some of the transcripts is shorter than the other ones, showing that there are some RNA degradation happening. So over the 3 years, we developed InTraSeq, which is basically an assay kit that comes with buffers that allows you to fix and permeabilize the cells and make sure that your RNA is retained inside the cell and that it's not degraded in the process. And because at CST we know how antibodies work, we found that once you conjugate these antibodies with a DNA sequence, you introduce a new variability to these antibodies, and we make sure that in the InTraSeq assay kit we have certain blocking reagents that guarantee that your antibody is specifically binding to your protein and that is not interfered by the DNA sequence conjugated on the antibody. So InTraSeq was developed and now it's commercially available online. It's short for intracellular protein and transcriptomic sequencing. I want to mention before I move forward that this technology was developed and validated by CST using the 10x Genomics Chromium single cell 3' kits with feature barcoding technology, and it's supposed to be used on the 10x Chromium instruments. Now, what is the InTraSeq technology? Basically, it's a 4-step immunostaining protocol, and it's an assay kit that comes with these buffers, and that will show you in the end of the talk. But once you have a single cell suspension, instead of going straight into, let's say, a 10x Chromium instrument and proceed with a single cell, you perform this 1-hour benchwork experiment, and that's split in these 4 steps. The first step is fixing your cells with methanol overnight. And at this point, we offer you the flexibility to store your cells for 7 days in the -20, or if you would like to store for a longer period, for 2 weeks, for instance, you could store them in the -80 freezer. And when you're ready to start, you proceed with this protocol and then continue with the single cell. Now, step 2 is where I would like to say where all the magic happens, because you have to mix different buffers in the InTraSeq assay kit that will make sure that from this point onward, your RNA is preserved in the cell. And also, we have these proprietary blocking buffers to make sure that your DNA conjugated antibody are specific and they will only bind to your protein of interest. Now, at this point, after the 30-minute incubation, you add the InTraSeq 3' cocktail, and we at CST understand that every antibody behaves differently. And certain antibodies require only a 30-minute incubation time, others require 2 hours. And we decided to do something unique, which you don't see with our competitors, and that is an overnight staining in the single cell protocol, because we want to make sure that the antibody signal is the best signal you could get, and you can get a very good signal to noise ratio from each antibody and for each target. And at this point, you wash your cells 3 times, which will take approximately 20 minutes, and then you start your single cell 3' kits experiment using the 10x Chromium. And this is a total of 1 hour of bench work spread out over 2.5 days. Now, in the next couple of slides, I'm going to show you the amount of information that you can get from an InTraSeq single cell experiment and show you the information that you've been missing. This is an example where we're showing you the UMAP, but on the left side of the UMAP, we're showing the RNA expression, whereas on the right side of the UMAP, we're focusing on the protein expression for the TCF1 target. And you can see that red means high expression, blue means low expression, and of course these are normalized values. You can see that the RNA is observed predominantly in this cluster. However, when you look at the TCF1 protein, you have a more robust staining condition, and you can accurately label TCF1 in that sample. Same thing here with NF81. You can notice that the RNA is overall upregulated in these three clusters compared to the cluster on the left. However, in the protein, you have an accurate representation where you have a gradient actually, where NF81 is predominantly expressed at the protein level in this cluster, and then at the protein level in that cluster is only in that region in this cell type, and so on. So with that data, what we're trying to say is that you get a uniform expression of your target using the InTraSeq protocol, and an accurate representation of that target expression that is observed at the protein level. And that could be due to many reasons. We know that in a cell, there are many more protein molecules in the cell compared to transcripts. That could be a reason why you have a more accurate representation of the target expression. And you could arguably say that protein is more stable than RNA, and that could be why you have a good uniform expression. Now, while we were doing and analyzing multiple data sets, we decided to hammer down and then understand the differences between RNA and protein. And what this graph is showing is the Pearson correlation coefficient, looking at the correlation between RNA and protein, and 1 would represent a high correlation, 0 would represent the lack of correlation, and a negative value would represent an inverse correlation. So you can see that with a lot of targets, and specifically surface proteins, there's a good correlation between RNA and protein. However, once you start looking at intracellular PTMs and post-translational modifications and intracellular proteins, that correlation drops significantly, and it drops very fast. And for instance here, when you look at, for example, phospho-CREB1 at the serine 133 residue, if you were to see high RNA levels in your single cell data, you honestly might as well expect low phosphorylation level of that protein. Now, we have to say that this result is not surprising, because we don't express post-translational modifications to correlate with RNA. But what we're trying to showcase here is that it's important to measure these signaling events in your single cell data, because you cannot infer them from measuring RNA alone. And it showcases how you can uncover missing information in your single cell RNA-seq experiment and offer new biological insights at the post-translational modification level. This is an example where we analyze a PBMC, peripheral blood mononuclear cell data set, and using the InTraSeq result, we found that CD8, which is a marker of T cell, is expressed in that region. So we took these T cells and then reanalyzed them using the single cell RNA-seq standard pipeline. And the standard pipeline, clusters the CD8 cell into 3 populations, which represent their differentiation states. So you have the CD8 naive cells, the memory, and the effector cells. So from there onwards, we wanted to see, like, when you recluster or reanalyze your cell of interest, how does the InTraSeq data set look like? And for instance, here we're showing TCF7 protein, which shows to be upregulated in the naive CD8 cell as opposed to the effector T cell. Now, this corroborates with a lot of previously published studies, and we were excited to see that it matches what's been published before. And when you look at the RNA, for example, you don't get the same expression pattern as the protein. And even if you were to sequence deeper, you will not get something as close as you were to get at the expression level of the protein. Now, here in the following UMAPs below, we were showing expression of TBX21, which is a transcription factor, and it's also upregulated in the differentiated effector T cell compared to the naive state. Same thing with phosphocrepserine 133 in that specific population. So using InTraSeq, you could analyze and categorize cell states in your data set that would sometimes be difficult when analyzing RNA alone. And you could use also, these signaling events to understand, different differentiation states within the same cell types. Now, in the next experiments, we wanted to see, okay, like, can we actually measure post-translational modifications? So we took Jurkat cells, which are cancerous T cells, and we inhibited them with wortmannin, and this is an acute inhibition of 30 minutes. Now, wortmannin is an inhibitor of PI3 kinase, and it's a kinase that is an intracellular protein that phosphorylates so many different proteins inside your cells. And in this UMAP, what we're showing is the drug-inhibited cells with wortmannin, and on the left side, you have the control cells that we're comparing the signaling to. So when you look at AKT protein level, you see that the protein is the same in both regions of the UMAP, in the drug-inhibited and the control cells. Now, at phospho-AKT, however, specifically at serine 473, you see that there's a downregulation of phospho-AKT only in the drug-inhibited cells with wortmannin, as opposed to the control cells. This is somewhat expected, and it's nice to see because PI3 kinase is upstream of AKT. So if the kinase is not working, it will not phosphorylate AKT. And that's exactly what we see in this data set, which offers a proof of concept that your protein is still there. However, your phosphorylation is downregulated. And because we know that this phosphorylation state of AKT reflects activity of your protein, you could finally measure whether your protein is functionally active or inactive in your data set. Now, S6 is downstream of AKT further down, and we looked at serine 235, 236 because we have a very robust antibody for that protein, and you can see that the signaling for phospho-S6 is downregulated in the inhibited cells compared to the control cells. And similar to the total AKT protein, the amount of S6 protein is still the same in both those two conditions. So finally, you can, in addition to RNA, you can measure your activation state of your protein and get some functional proteomics insight in your single cell RNA sequencing data. Now, you can imagine that things can get very complicated and very fast too. And this is what we're showing here in just one UMAP, where we took PBMC cells, the peripheral blood mononuclear cells, and we treated them with lipopolysaccharide, which is represented in the green square, and compared them to the untreated cells. Now, this is a chronic stimulation for 3 days, and what we're showing here is phosphostat-3 serine 727 expression in those conditions before and after treatment. So that expression is observed in this row. White represents low phosphorylation levels, red means high, represents high phosphorylation levels. And you can see that with changes in treatment, once you treat the cells for 3 days, let's say in these two columns, there's an increase also in phosphostat-3, and that's also correlated with certain transcripts or protein that change expression in after treatment. And you can also see that some targets were downregulated. And since you're leveraging a single cell RNA-seq platform, like I mentioned in my second slide, it's a technology that's been used to look at the heterogeneity in the cells. You can correlate the signaling levels with transcript in many different cell types. And in this case, like CD4, untreated, treated, or monocyte, untreated, and treated cells. So you can finally gain a comprehensive understanding of signaling pathways at the single cell resolution, where you can measure the phosphorylation level of certain proteins, and you can understand how the transcriptome is impacted in individual cells. Now, in the next couple of slides, I'm going to talk about the bioRxiv manuscript that we have listed. Please feel free to check it out. It's available online, and it's currently under review, and we're addressing the reviews. And like I mentioned earlier, it's a collaboration with Dr. Vijay Kushru at Brigham Women's Hospital at Harvard Medical School. And I'm working with fantastic postdocs in his lab, Ling Lin and Zhao Kai. They're very smart people and technically gifted. So in today's webinar, I'm going to go over some of the data in Figure 3 and some of the data in Figure 4. But if you're interested in reading more about this, check it out in the bioRxiv. It's currently online. So when we started this collaboration, we wanted to understand how robust is the signaling pathway with InTraSeq. So traditionally, when you measure phosphorylation events, it's done on a Western blot experiment, where here, for instance, when you look at NF-kappa B, phospho-NF-kappa B at serine 536, we took naive T cells and we compared them to different stimulation levels for 10 minutes. And we have the PMA as a positive control. We have pathogenic Th17, non-pathogenic Th0, and the naive, like I mentioned earlier. And we find that phospho-NF-kappa B is upregulated in the PMA compared to the remaining conditions. Now, I want to mention that we did a biological replicate and then performed InTraSeq. So this experiment was done on a different day on a different set of mice because we isolated primary T cells, and done by a different postdoc also compared to the Western blot. And you can see nicely that, you know, in this dot plot experiment and the graph, the phospho-NF-kappa B is low in those 4 conditions and it's upregulated in the PMA, similarly to the Western blot. And we're very pleased to see that, which tells us that, you know, even though it's a biological replicate, we were able to get the same exact data compared to the Western blot. Now, granted, in the InTraSeq experiment, we combined all the cells together and we averaged out the phospho-NF-kappa B. So this is a simplification of the InTraSeq data set, and it's actually way more complex than this. But we wanted to make sure that this dot plot would be comparable visually to the Western blot. And of course, these are different experiments. You know, the Western blot is a chemiluminescent assay, whereas the InTraSeq is a sequencing assay. So there are some minor variabilities if you squint a little bit, but the main patterns are preserved between those 2 experiments. Now, we also noticed in this experiment that STAT3, specifically at phosphorylation at the tyrosine 705, and that this signaling is specific to the pathogenic and non-pathogenic conditions compared to the remaining 3. And with the InTraSeq experiment, we saw the same patterns here. Now, you know, if you want to look at the data set in the UMAP, that's how it would look like. Phosphostat3 tyrosine 705 is predominantly upregulated in those 2 conditions compared to the 3, which is interesting. And we started to dig deeper into that data set and understand what's happening in those 2 subtypes. So as a quick summary of the experiment, we isolated different lymph nodes from the mice and then sorted naive T cells using, you know, CD4, CD25, CD62, and CD44. And then once we isolated these naive CD4 T cells, we polarized them to become Th0, non-pathogenic Th17, pathogenic Th17, and we also have a positive control with PMA and ionomycin, which is a very strong stimulant that will induce signaling in your cells. And we did that in different time points, 10 minutes, 45, 6 minutes, and 24 hours. Now, we don't have the PMA treatment for these 2 conditions because it's a very toxic system. And then after, you know, an hour, the cells started dying. But what we want to show in this experiment in the UMAP, I want to focus on this region, which shows the naive 0-minute cells or the 10-minute stimulated cells. So we're showing the time points, not the clusters. And we see that these 2 time points are in the same region of the UMAP. And like I mentioned earlier in my talk, the closer the cells are in the UMAP, the more similar they are transcriptionally. And, you know, that's somewhat expected because 10 minutes is not enough to drive the transcriptional profile to change in your cells. You know, when, let's say you're stimulating your cells, your signaling pathway is going to be changing, and, you know, once that happens, you recruit transcription factors to promoter regions, and then these transcription factors promote polymerases, and then they induce transcription. That process takes time. It takes at least, you know, 30 to 40 minutes for something to happen. And in that, you know, 0-10 minute time point, you can see that this is the UMAP on the right, showing the different sample types. You can see that they all cluster in the same region as well. And this is done, you know, based on the standard analysis where you're clustering cells based on, you know, over 1,000 differentially expressed genes, and it's not enough to find these changes. And like I said, biologically speaking, it's not enough. However, because in the InTraSeq cocktail, we have a lot of antibodies that measure signaling events, if you were to cluster your cells based on these proteins, you get a completely different story. And that's what we're showing here. So this is the same UMAP as the one above, but we're clustering the cells based on the protein and the antibodies in the cocktail. And you can see that at 0 minutes and 10 minutes, you have 3 different clusters at least, showing that you can finally observe and understand these early events in your single cell data sets that you were missing at the RNA level. So in this example, this is the same UMAP. You can see that the naive cells are in this region and that region, the pathogenic in that region, and then you have a PMA sample. But either way, like these events, since you can finally understand them at early time points, it can help you understand why transcriptionally at 24 hours, you have these 3 distinct clusters, because we do see them in the InTraSeq data. You have these early events that you can finally measure. So this was done only by clustering the cells based on 30 targets. And as we're expanding the number of antibodies in our InTraSeq pipeline, hopefully we'll get even a better separation, especially at earlier time points that you were missing at the RNA level. Now, one of the changes that we found was phospho-STAT3, and like I showed you earlier, and this is showing the normalized expression between the different cells for phospho-STAT3, and in this case, it's tyrosine 705. So focusing on these 2 samples, the non-pathogenic Th17 versus pathogenic Th17, you can see that there's an upregulation in phospho-STAT3 compared to the other conditions, and specifically here Th0. So we decided to look at CITE-seq data set for STAT3 that is publicly available, because STAT3 is a molecule that once it's phosphorylated, it translocates to the nucleus and it acts as a transcriptional activator, and it recruits other proteins to promoter regions. And there's, the promoter regions that STAT3 is bound to are very well known in the field. So we focused only on these STAT3 bound genes, and we leveraged the single cell RNA data and decided to look at the RNA expression of these genes. And when you look at non-pathogenic Th17 compared to Th0, the STAT3 bound genes are gradually differentially expressed as time goes on, at 45 minutes, 6 hours, and 24 hours. That's what it's showing. Some genes were upregulated, some genes were downregulated, showing that not only is the signaling event increased in these 2 samples, you see that the transcriptional machinery is also differentially expressed. Now, in this data set, we found that at the 24-hour time points, in both phosphorylation events for STAT3, whether it's serine 727 or tyrosine 705, you see a downregulation of that signaling in pathogenic compared to non-pathogenic T cells, T17 cells. And we wanted to see whether that difference is actually real. And we did the same analysis where we looked at the RNA expression of the STAT3 bound genes between non-pathogenic and pathogenic. And you can see that in the first time points, there is no difference. However, suddenly at the 24-hour time point, it goes from almost 0 to 700 differentially expressed genes, whether they're upregulated or downregulated, hinting that that change in signaling is likely to be real in STAT3. Now, here on the right side, we're showing a heat map focusing on the 24-hour time points, looking at the RNA expression between those 3 samples, the Th0, non-pathogenic, and pathogenic. It's just a visual representation that shows that there's indeed certain RNA transcripts that are upregulated in the non-pathogenic compared to Th0 and in the pathogenic sample. And because we have certain targets that are already in the InTraSeq panel, we cross-validating that change in the protein level also using our InTraSeq panel. And we performed qPCR, and we found that, for instance, GMAP5 is indeed actually upregulated in the pathogenic compared to non-pathogenic using, like I said, the qPCR data set. And it was, very reproducible. We had 4 replicates per target. So what we're trying to showcase here in this schematic, that even though in non-pathogenic T cells and pathogenic Th17 cells, in both cases you have an increase in STAT3 signaling pathway. but it's not fair to assume that that signaling event is going to behave similarly in those two different cell types. Now, STAT3 binds to a lot of different promoter regions in the genomes. You know, it's fair to assume that certain targets are going to be upregulated in one cell type and not another cell type, and that's exactly what we're showcasing here. So STAT3 gets phosphorylated, it translocates to the nucleus. FOXO1 is upregulated, so is MAF, and CD200 is downregulated in the non-pathogenic Th17 cells. However, in the pathogenic Th17 cells, you have a different story where LIF, PIM1, SADB1 is upregulated, whereas ILOS is downregulated in that same cell type. Check out the manuscript if you're interested in learning more about the signaling pathway, but this is showing how you can quantify, you know, your protein, your PTM, your RNA, all in one experiment, and you can understand the molecular mechanism of, you know, differentiation and whatever biological system you're working on. Now, traditionally speaking, in single cell RNA sequencing data sets, there are standard pipelines to look at cell cycle phases based on the RNA expression. So we took here the TH0 cells at the 24-hour time point, and, you know, using the standard cell cycle phase scoring, you can see that there are three phase calls. You know, the G1 in gray, the G2M in blue, and then the S phase in yellow. Now, and these are the different percentage of cells in the sample. You know, 28% are in the S phase, 64 on the G1, and so on. But you notice that, you know, the G2M are combined together, and, you know, the cell cycle phase, the cell cycle is technically 4 different phases. You know, it's the M phase, which is mitosis, is a distinct phase in cell cycle, but transcriptionally, it's very difficult to label the mitotically active cells in a single cell RNA sequencing data set. And that could be arguably because mitosis is such a short phase compared to the remaining cell cycle and compared to the interphase phases. And it's traditionally, you know, on average, it depends on your cells. If they are dividing, it could be 1 hour. And if your cell is dividing here, in this case, like, you know, 24 hours, you know, 23 out of 24 are in the interphase. Now, at CST, we have a very robust phospho-histone 3 serine 10 antibody. And I'm showing you here images from our website. This antibody specifically labels mitotically active cells. And, you know, in the interphase, it's not observed at all, but then in mitotically active cells, like here in the anaphase or in the metaphase, you see that in green, this phospho-histone serine 10 is upregulated only in the dividing and the mitotically active cells. And we used that antibody to see how it's expressed in the cell cycle phase scoring. So we found that phospho-histone serine 10 is upregulated in this region of the UMAP as well as that region. And, you know, going back to the data above, in this region, these are the G2M phase, which is nice to see because some of them are indeed phospho-histone 10, serine 10 phosphorylated, which they are actually mitotically active cells. But we also noticed that in this region of the UMAP, these are also mitotically active cells based on that signaling event, and it's predominantly based on the RNA expression, at least, labeled as G1. Now, this is not surprising because, you know, mitosis is flanked by the G1 and the G2 phases. So we do expect, you know, if based on the protein level and you know, because the clustering here and the annotation was done based on the RNA, that we do, it makes sense that the mitotically active cells are observed also in the G1 phase. What's nice to see that in the S phase, which is the furthest away cell cycle phase compared to mitosis, you don't see any expression of phosphoserine 10 here. So using a newer annotation that we have in the manuscript and an algorithm that we wrote, you can finally accurately label mitotically active cells. And if you're interested in, you know, dividing cells or you know, if you study cancer biology, you can accurately understand whether your cells are arrested at the G1, G2 phase specifically, or, you know, if the mitotic phase is, you know, is longer and so on. Now, we do get a lot of questions, especially about multiplexing and hashing your samples with InTraSeq. And I want to tell you that starting today, you can finally do that. And this is what we're showing here. So I'm taking a snap of the protocol, one section, where you mix the antibody master mix in the InTraSeq protocol. And the assay kit comes with a positive control, the histone H3, and it comes with a negative control. And we decided to leverage the positive control and allow you to use that so you can multiplex and hash your samples. So instead of using the histone H3 antibody that is already available in the assay kit, you can now use any other histone H3 conjugates, and we released 3 additional histone conjugates in today that I will talk about in the next coming slides. But you can label that cell with a different conjugate, and I will show you in the next couple of slides what that means. So assuming that this sample 1, you process them with the InTraSeq protocol, this sample 1 was labeled with the histone H3 conjugate 3048, which has a unique DNA sequence and a DNA barcode. You do the InTraSeq with that histone antibody. However, in parallel, you could take sample 2 and then process them with the InTraSeq protocol. However, instead of using this conjugate, use another histone conjugate with a different DNA barcode, and then so on with sample 3 and sample 4. And then when you're ready, when you finish the InTraSeq experiment, you can pull and mix your samples together and then process them in a single cell experiment. This way you can save your cost on your single cell reagents. So this is an example here, what we're trying to show. The antibody binds to the chromatin region, the histone H3. This one has a unique barcode, whereas that histone H3 has a different barcode. They're color coded to show that they are different, basically. So in this UMAP, we're showing the 2 different samples. These are sample 1 are cultured T cells, sample 2 are PBMC cells. They're transcriptionally different. We decided to use these 2 cells because they're easy to visualize on the UMAP. And sample 1 is labeled with 1 histone, and you can see that very nicely here with the histone H3 conjugate 3048. So you can see that the expression is very specific to sample 1, and you don't see any expression in sample 2. Now, sample 2, however, was labeled with a different histone here, with the histone 3049. You can see that it's specific and it's upregulated only in that sample compared to the rest. Now, in this case, these 2 samples are transcriptionally different, but sometimes if you're using the same cell types that are stimulated, or there's different, you wouldn't be able to tell them apart because they would cluster together if they're transcriptionally similar. However, using our histone conjugates as hashing reagents, you can demultiplex them after you sequence, because you know that this sample only has this barcode, and then this sample also has another barcode. And this is another example, in that same sequencing lane, we also have sample 3, sample 4. It's the same thing that we're showing here. Sample 3 was labeled with the histone conjugate 3002, and then sample 4 was labeled with the conjugate 3050. Now, this is a violin plot to show the same data set as I showed you in the previous slide. And you can see that histone H3 3048 is very specific and unique to sample 1. We don't see any leakage or any expression in the other samples. So you can accurately label and computationally isolate each sample, and hopefully save on your costs in the single cell experiment. Now, we also got this question multiple times, like, how is this different to surface hatching? So in that experiment, actually, we labeled sample 1 with with antibodies that recognize surface proteins. And we found surprisingly that these antibodies have some expression in sample 2 when it shouldn't. So we do see a strong signal in sample 1 as expected, but you do see some background as opposed to our histone H3 conjugate, which is very specific. So hopefully, we hope that if you were to use these histone conjugate, you can get a better staining and a signal to noise ratio in your single cell experiment, so you can accurately label which cell comes from which sample compared to surface hashing. We've done that also again in another sample, and we see the same pattern with the surface protein hashing. Now, some of the product offerings we have here, we have 38 individual. However, because today we just released 3 additional more, there's actually 41 conjugates. And we have currently a cocktail 1 that has 31 antibodies in the cocktail. And this cocktail one is both human and mouse reactive. And you can see that there are 7 antibodies that are not in the cocktail, and these antibodies are only human reactive. And now the assay kit is, like I mentioned, has all the buffer components that allows you to perform the InTraSeq experiment. And it comes also with a positive control, one of the histones, and a negative control, a rabbit isotype control that doesn't bind to anything. So if you're interested in normalizing your data set to these 2 targets, you could also do that as well. This is what we have currently in cocktail 1. And you can see that, these are color coded, but we have a lot of antibodies that are specific for post-translational modifications, whether it's Phospho-Histone serine 10 that I mentioned in the earlier slides, and we have 2 histone marks for condensed and loose chromatin, phospho-S6 for EBP, phospho-CREB, phospho-ERK12, and then phospho-AKT, like I showed you in the earlier slide, and the STAT3s. We have 2 phosphorylation antibodies for STAT3, the tyrosine 705 and 727, and it's nice to see the interplay between those 2 phosphorylation marks and STAT1 and NF-kappa B, as well as glucocorticoid receptor. And stay tuned, because in the next coming weeks, we're releasing cocktail 2, and if you do a lot of cancer research, I believe you're going to be very excited in the next cohort of cocktail 2, which is going to be specific to the cancer field, as well as the epigenetics field as well. And like I mentioned, some antibodies are not in the cocktail 1, and these are only human reactive. But if you're interested in TBET or TBX21, it's a very strong antibody. It gives a very specific signal in the InTraSeq dataset if you're interested in using it. We also have S100A9, CD68 to label macrophages, and we have T-cell markers. We have an additional isotype control if you're interested in adding more than just one control in your experiment. Now, quick information before I end the webinar, that if your favorite antibody is not conjugated currently, we do offer a custom conjugation service. So you can go in on the website and then go on the service and then the custom conjugation. And we can conjugate any of our antibodies with a 10x feature barcode. However, we will not validate them in the InTraSeq assay, but I will help you determine the most optimal concentration of that antibody in the InTraSeq assay. And because our protocol is very flexible, you can add and mix any antibody that you would like. I will just have to work with you individually and personalize it so that your antibody is at the proper concentration, so it's not taking a lot of your reads in your sequencing. Now, we also recommend you to have more than 1 sample per experiment. And that's because a lot of our antibodies in our cocktail are for measuring post-translational modifications. And these proteins are ubiquitous traditionally in your cells. So if you want to get the maximum amount of data in your sample, make sure you have the different conditions, whether you have control stimulated cells or knockout cells or overexpressing cells. You have things you can compare them to. And like I said, we want you to maximize the information from your signaling pathway in your single cell data set. Thank you so much for attending and being patient with me. And we have the world's most trusted antibodies, and I will be happy to take any question you have today.
Thanks, Maj, for a great presentation. We do have a couple of questions. So the first one is, methanol is harsh on cells. Doesn't it negatively impact surface proteins?
That's a good question. So methanol does impact surface protein structures. And this is why in the InTraSeq validation at CST, we make sure that the antibodies that are commercially available have been tested and validated in methanol. So for instance, like surface markers like CD4 and CD3, we make sure that even though the protein is slightly denatured, it will still bind to that protein in methanol fixed cells. Yeah.
Okay. How many cells should people start with?
So in the protocol, we have, in the first step of the protocol, it tells you to start either between 1 to 5 million cells, and that varies on purpose because we find that if you're working with primary cells, if you start with less than 1 million cells, we want to make sure that you have enough cells at the end of the assay because there are so many centrifugation steps. And usually, if you start with 1 million cells, it should be enough for you to perform around 6 different single cell experiments. But if you're working with cell lines, you can start on the low end and that wouldn't be any issue. But it's between 1 million to 5 million cells.
Okay. And then can I combine multiple samples in one experiment before running InTraSeq?
Oh yeah, that's what we released today. So we have these hashing reagents, the histone antibodies that we just released today, and please check our website. These are also both human and mouse reactive, similar to the histone conjugate in the InTraSeq assay kit. So you could label your cell with another histone conjugate and then combine multiple samples, and then after you sequence, you can demultiplex them so you can understand which samples comes from where, basically through the histone barcodes. Yeah.
Okay. I think that was our last question. If anybody has further questions, you can check out our website or reach us at support@cellsignal.com. We'll get you in touch with Maj to discuss your experiment or what you're thinking of. But our time is now up, so thank you for joining us today, and we hope you enjoyed the presentation. Thanks again, and have a great rest of your day.
Thank you everyone for joining in, and have a good rest of the week. Bye-bye.